Okay. Hello, AP Bio kids. Welcome to review number 1. And this is kind of going to be the setup. So I've got the notes over here to the right. I've got a whiteboard, a makeshift whiteboard in Microsoft Paint here to the left. And we'll see how this goes. Now, this is going to be the overview of the first unit. Right now, FRQ number 2 is posted. It should follow along with this unit, specifically what I think they're going to ask you related to this stuff, what they could ask. They've said that they're not going to ask you anything like content related. So I'm still going to give you the information just in case you need to know it, but the free response is going to cover what I think they're going to ask you, which is mostly going to be enzymes. So right here on my paint, this is really for me, but it also kind of gives you an idea about what we're going to be talking about today. So we're going to start off with water and bonding and properties of water and then all that kind of stuff. And then we're going to get into biomolecules, and then we'll finish off with enzymes. And I'll try to go as quickly as possible to make this as watchable. Okay. All right. So let's start off. Now, notice here on the right, I've got this chart, this periodic table, and what you'll notice is this is based on electronegativity, okay, which is basically how much different elements want to attract to one another. Okay, so we can see the farther right on the periodic table we go, the more electronegative we get. It means those elements want to attract more to other elements. Okay, they want to gain electrons. These guys over here that have a low electronegativity want to give electrons. Okay, so how does that relate to water? Well, the structure of water, we know we have an O, and then we have two hydrogens. I was thinking this earlier. It's kind of sad that as I'm writing with the mouse and paint, this is actually arguably better than I would write hand. Anyway, so we've got our water molecule here, H2O. Okay. And you'll notice that oxygen has a higher electronegativity than our hydrogen does. We're sitting here at 3.5 for the oxygen, and the hydrogens are at 2.1. So what that means is that the oxygen is slightly more negative than the hydrogens, which means it says the hydrogen has a more positive charge, the oxygen has a more negative charge, okay? So what that means is that electrons spend more time around the oxygen. We're going to have more electrons around the oxygen over time than we do the hydrogens, okay? Now what this creates is what's called a polarity, where overall water has a neutral charge, okay? But this top side of the water molecule is more negative, and this bottom side of the water molecule is more positive. And this is what allows water to bond to itself and to other things. So this difference in charge is what's called polarity. I'll just write polar. And because this end is more positive, when we get another water molecule here, it's the negative end is going to want to bond with the positive end of the other one. Okay? So here we notice that we have the negative end of the oxygen on this water molecule is trying to bond with the positive end of the water molecule on the top. And so we get this bonding, which I'm just going to draw with a big old line. Okay? Now, this is what allows water to bond with itself. That is what is called cohesion. Okay? Cohesion is water bonding to itself. It's also what allows adhesion, which is water bonding to other things. So the fact that water is polar, where we have a negative charge on one side and a positive charge on the other side, is what allows water to bond to itself and to other things. Okay, the main place we're going to see this in nature, and where they might ask it to you on a test, is through plants and transpiration. Okay, now you'll notice that water, because it can bind to itself and because it can bind to other things, it allows it to sort of defy gravity, and that's a really important adaptation that plants have taken advantage of. Even in recorded videos, I still can't find the damn picture I'm looking for. That sucks. Okay, well, so in a water molecule, transpiration. I'll just pull up good old handy dandy Google here. Okay, so we'll notice that in a water molecule, it rains, it's going to go, that water molecule is going to go into the ground, and then as this plant is consuming the water is going to travel up from the roots to the leaves of the plant where it's doing photosynthesis. What allows for that to happen is the cohesive and adhesive properties of the water. So essentially what happens is in the tree, can we see this tree? Is this tree making it? No, it's not. Okay, I'll just do it down here. There we go. In this tree, look at that tree. Dang. I should teach freaking AP art. I'm so good. Okay, so you've got the water molecule right here, right? As it goes up, it's the adhesive properties are what allowing the water to stick to the inside roots of the plant. So it's sort of sticking to the sides of the plant, and then it's sort of bouncing up, and that's how it's traveling up. And as it's going upward, the cohesive properties are what are bringing other water molecules with it. So they might ask you something about how do plants take advantage of different properties of water. It's taking advantage of adhesion and cohesion right there in order to get the water from the roots to the leaves of the plant. Now, what's fueling that to happen is that leaves in plants have these pores called they're called pores, and every morning, plants open those pores to let water out, and that's why there's morning dew. And so what's happening is as that water is leaving and evaporating and going into the sky, that's sort of creating a suction and treats the stem of the plant like a straw, which sucks the water up. So this is the adhesive and cohesive properties that are allowing plants to take advantage of water. Now, this bond that we created, this is what is called a hydrogen bond. Okay, now hydrogen bonds are very weak bonds. There we go. And you'll notice hydrogen here, we thought only hydrogen can make one bond. That's what we learned in chemistry. Well, it can only make one covalent bond, which it's making with the oxygen. Okay, this is a covalent bond. The bond that it's making with the other water molecule is a hydrogen bond. Okay, now these are very weak bonds that can be broken really easily, which is why you can break water. It's why you can swim and cannonball and all that stuff. But it's what allows water to sort of stick together, giving it the adhesive and cohesive properties. Okay, so that's the first kind of bond, is the hydrogen bond. Now, let's go on to something else. Bye-bye. Let's talk about some other molecules. So let's say I've got two ring structures here. Okay. Now this one, I've got a, let's undo that. So let's say this one has a hydrogen coming off of it. This one's got a hydrogen coming off, and it's got an alcohol group. Let's say this one's got an alcohol group too. Okay. Now, if you remember, I've got two molecules here. Now, if you remember, both of these molecules are rings, which makes them both carbs. These are carbohydrates, and we know that because they are in the shapes of rings. Okay, that means they're sugars. Now, if I want to bond these two molecules together, I have to take advantage of water. And how I'm going to do that is I'm going to just draw a line connecting them. But in order to draw that line, you notice that at the point we have a carbon here and we have a carbon here. Carbon can make 4 bonds. Okay. Now, if I count, I've got one bond coming off here. I've got one bond coming off here. I've got one bond going to this hydrogen that is poorly drawn up here at the top. And I've got one bond coming down here to the alcohol group. So that's 4 bonds. So if I want to make this 5th bond, I'm going to have to get rid of one of the other bonds. So what I can do is I can take the alcohol group here, and I can take the hydrogen here, and I can remove them. And when I take a hydrogen and combine it with a oxygen and another hydrogen, I get H2O. So by removing the water, I create that bond. Okay, that is what is called dehydration synthesis. Okay, we are dehydrating because we're removing water from the top, and we are synthesizing this new bond. So by removing water, we are creating a new bond. Okay. That is dehydration synthesis. The opposite is hydrolysis, and that's where I just take water and I add it back in. So let's say I'll just draw an arrow going right back. So I take this water molecule and I split it up. I add in one hydrogen to this side. I add in the alcohol to this side. And now it takes that bond that I just made and it goes goodbye. And now we have two individual carbohydrates, okay, or two individual monosaccharides. So water is really important to us to ingest in our bodies because we need that water to break down and build molecules. I mean, your body, when you eat food, is just constantly taking these large things, breaking them down into small pieces, and then building them back up into what you need, okay? We're going to talk about that a little bit more here in a second when we talk about proteins, okay? But that's how proteins work. We're just taking proteins in food, breaking it down into individual amino acids, and then recombining those into the different molecules that we actually need, okay? Now, I'm trying to go quickly because of time. And this is meant to just be sort of a review. But I also want to be as encompassing as possible. So, all right, so let's go into the next thing, which is fats. Now, here on your screen, we've got a fat. Okay, now remember, we have two kinds of fats. We have the saturated fat. That is not the writing tool that I want. We have a saturated fat, and we have an unsaturated fat. Okay, now here we have three fatty acid chains. These top two, this one, can I do that? Oh, look at that. This one and this one are saturated fats. Let's just highlight both of them. These two are saturated fats, and we know they're saturated fats because they are straight. They're in a straight line. And saturated fats are always in a straight line. This one down here, this is an unsaturated fat, and we know that because there's this double bond right here that causes this kink, okay? And that kick out is what makes this fat an unsaturated fat, okay? We call these saturated fats because they're straight. It means we can basically pack more of them in closer spaces. Okay, so saturated fats are what were long believed to be bad for you because of that reason. Because we thought that you could pack more of those fats close together, which would be bad for something like clogging your arteries. Right? Saturated fats also become solid at room temperature. If you think about like bacon fat or animal fat or butter, if you leave it out, right, it turns into a solid. Imagine that happening within your arteries, and that's how you get heart attacks, okay? So these saturated fats are generally bad for that reason. These unsaturated fats, because they have this kink, it means that they can't solidify. So even if you leave them out, they turn into a liquid. So that's why people think that unsaturated fats are not as bad for you as these saturated fats, okay? But honestly, this is all, the thing with fats and carbs is a lot of the stuff they could ask you is just knowledge-based information that you could Google on the test. So really the only thing that I think that you should know about carbs and fats is that they both have the same function, which is to store energy. Okay, that's all they are used for. Fats are more for long-term energy. Carbs are more for quick sugar, short-term energy. That's why you get the sugar rush. But both of them do the same thing in that they're both for energy storage, and you both need them for energy. Okay, carbs are more for energy you need right now. Fats is more long-term. Okay, but otherwise, that's really all you need to know about carbs and fats is they're both for energy storage. Okay, what they could ask you more information on is proteins. So let's get into proteins. Now remember, where's my picture of a protein? Okay, so here we have a couple different proteins. Okay, now you'll notice that every protein. Remember, we studied that proteins have four basic parts. They have first they have a central carbon, and then they have a carboxyl group. They have a hydrogen group. They have the amine group, which is the nitrogen with two hydrogens coming off of it. And then they have the R group. And the R group is sort of a placeholder. It's like a variable in math. It's like an X, Okay, it could be anything. And so you can see here, we have all these different R groups. So here's a CH2 with an alcohol. Here is a CH2 with a 6-carbon ring and an alcohol. Here's a sulfur group. Here's a CH2 and another alcohol, okay? So we have all these different R groups, and these are what we call amino acids. So these, this whole thing is an amino acid, and there are 20 different amino acids because we have 20 different R groups. Now, these are the R groups, and what you don't have to know anything about them other than there's 20 of them, and what's different about them is their shape. Because remember, with proteins, shape is everything. Because what we're going to talk about here in a second is the shape of these proteins determines how they connect together, which gives the property of the protein. Okay, whether it's hydrophobic or hydrophilic, whether it can stand water, whether it's in the cell membrane, whether it's inside the cell, whether it can be out extracellular. All those different properties have to do with the R groups that are in those amino acid chains. Okay, so we've got 4 different parts of our amino acid. Okay, we've got these 20 different R groups, which give us 20 different amino acids. Now, in this picture, what you're seeing is if I want to take 2 amino acids and I want to combine them together, what I'm going to do is I'm going to use the process we just did, dehydration synthesis, where I'm pulling out a water molecule. And by doing so, I combine the carboxyl group of 1. Oh, I did that wrong, didn't I? There we go. We take the carboxyl group of 1 amino acid and we combine it with the amine group of another amino acid. And that's where we get this bond right here. Now, this is a special type of covalent bond called a peptide bond. A peptide bond. And it's a bond that's just a covalent bond, but specifically for proteins, okay? So here, when we have multiple amino acids that are bound together, that's what we call a polypeptide. And I kind of feel bad that I'm writing this terribly. I guess it's no different than normal. That's just a character flaw. I'm just, borderline illiterate. That's okay. I hope you can understand at least what I'm saying if you're actually paying attention and not like listening to music or something. This is a polypeptide, okay, where we have multiple different peptides, multiple different amino acids that are bound together in a chain, okay? Cool. Now, here's where the R group comes into play. So you'll notice here we have all these different R groups. Some of these are bigger, some of these are smaller, some have more stuff, some have less stuff, okay? Now where this comes in order is once we get this protein, we sort of have to start folding it, okay? And I wish I had a... Oh, God. Oh, gee. Oh, good Lord. Things are spilling. Okay. All right. So here's my makeshift example. So let's say I have this cord, okay? Let's say this cord is my polypeptide chain, okay? This is, if you remember, this is what we call our, oh, that's not what I wanted to do. This is what we call our primary structure, okay? Your primary structure is just your amino acids in a chain. Okay? So this is my primary structure. This is my amino acid chain. Okay? Just kidding. This is a USB cable, but use your imagination. This is my amino acid chain. Okay? Now, once I've got this amino acid chain, what I want to do is I want to start folding it because I want to condense it into the actual shape that it's supposed to be. Okay? That's what we call our secondary structure. And our secondary structure, remember, we had 2 different kinds of secondary structure, and that's what we have right here. So here's my primary structure. Here's my chain of amino acids. My secondary structure, we had 2. We had the beta sheets, and we had the alpha helix. I don't know why I did the B one first before the A one. I hope for you OCD people that's just bothering the crap out of you. That's kind of on purpose. Let's just imagine I did that on purpose. So we have the beta sheet, we have the alpha helix. Okay, so what I can do with this chain is I can start to, in the case of the beta sheet, I can just start to wrap it. Okay, I can start to bend it, fold it, sort of like this. Okay, this would be an example of a beta sheet where it's just sort of wound back and forth. Okay, an alpha helix is a little bit different. An alpha helix is if I were to were to wrap it, okay, that would be an alpha helix. So I can take my structure and I can bend it those two different ways. Now, where bonding comes back into play here is that as I start to wrap it in the form of a beta sheet or in the form of an alpha helix, I start to use hydrogen bonds again. So here we have the hydrogen bonds coming back into play, because what's going to happen is based on my R groups, I'm going to get hydrogen bonds between my coils of our alpha helix or between the folds of our beta sheet. And what this is going to do is it's going to provide some structure to our protein. It's going to make them a little bit more sturdy and not as likely to just like break down and go back into our chain original primary structure. Okay. So here we have a hydrogen bonds come back into play. Okay. Now, then our next form of structure is our tertiary structure, and this is where we start combining our beta sheets and our alpha helices. So maybe we have a beta sheet over here, and then we go into an alpha helix, and then we have another beta sheet, and then we have another alpha helix, and then we have another beta sheet, and then an alpha helix. Okay, so we start to combine those two things together. Okay. Now, as we start to combine those things, we start to see our other kinds of bonds. We start to see ionic bonds start to form, okay, in the form of like disulfide bridges. We start to see our covalent bonds start to form, where we have different covalent and ionic bonds, where we have different R groups that are bonding together, okay, as well as the hydrogen bonds that we had in our secondary structure, okay. And then our last one is our quaternary structure, which is where we just basically take this sort of like group of mesh. So here I've got my beta sheet and my alpha helix, and then I just sort of like group it together. Okay, well now this is a tertiary structure. So if I had another one, which I don't have another one. Imagine I had another one. And I compound them together. So now I have two things. Those two things, this is now a quaternary structure. This is a full protein, okay? So a quaternary structure is just when I get multiples of these together in a group. That is my quaternary structure. I don't like that. I don't like that. Let's see if I can. Dude, drawing with a mouse is freaking hard, man. Okay, so now I've got a quaternary structure, okay? Which here kind of looks like this weird mass of brain-looking stuff, okay? Same thing, all right? So we have our primary structure, our individual chains of amino acids. Our secondary structure is our beta sheets and our alpha helices. Our tertiary structure is when we start to combine beta sheets and alpha helices into one string. Notice that these are all continuous, right? It's one long string that just coils and bends and coils and bends. And then when we take those individual components and start to group them together, now so our quaternary structure is like 4 different segments, okay? Or 3 different segments, or whatever it has to be, whatever number, okay? Now, the quaternary structure is where we start to get our van der Waals interactions, okay? And this is where we start to get our protein structure based on hydrophobic or hydrophilic. Good Lord, this is difficult. Interactions. So the way that our proteins fold isn't just out of sheer happenstance, just randomness, right? It's folding specifically based on whether these R groups like water or do not like water, whether they stick to one another or they don't stick to one another. So the order of our amino acids is going to determine how it folds, and it's also going to determine what parts are on the inside, on the inner part of our protein, and what parts are on the outside. Our hydrophilic parts are going to be on the outside because they're cool with water. Our hydrophobic parts are going to be on the inside. Good Lord, I said that wrong. Hydrophilic parts are going to be on the outside because they're cool with water. Hydrophobic parts are going to be on the inside because they're not cool with water. Okay? Man, I'm really good at this. I should get paid to do it. I'm just kidding. I'm really terrible. Anyway. Okay, so we've done protein structure, R groups, polypeptides, primary quaternary structure. Okay, now, how does all this fit together? And this is where we get into the nuts and bolts, because this is where I think if they're going to ask you something about all this kind of crap, this is going to be how they do it. And it's going to be in the form of enzymes, okay? Because enzymes, I'm going to do that a little bit smaller up at the top. Enzymes are a type of protein, okay? Enzymes are a type of protein. Remember that enzymes always end in A's. Okay, that's how you know an enzyme. And for further learning, most proteins either end in A's or in. Okay, so if you ever see something that you have no idea what it is, but it ends in IN or it ends in EN, that is probably going to be a protein. Okay, just so you know. That's going to be important because on one of those two FRQs, they're probably going to throw some crap at you that you've never seen before. But if you look at the ending, you can probably maybe figure out what it is. Okay. All right. So we've got these enzymes, which are a protein. Okay. Which means all the stuff we just said about proteins apply to enzymes. Okay. So they're made of amino acids. They have different R groups. They're shaped a certain way. So maybe we have this mesh of stuff that forms a specific shape. Okay. So this is my protein that I'm forming into a specific U shape, and this protein is now an enzyme, okay? Well, remember that our enzyme has what's called an active site. So an active site, okay, I'm not going to write as. I actually have to spell it out, damn. Active site. Okay. Now, the active site is where our substrate is going to bind to our active site. And remember that our substrate, do this in green, our substrate has to specifically fit the enzyme, which you wouldn't be able to tell from my really crappy picture here, but the substrate has to fit specifically for an enzyme. It's like a lock and a key, okay? So we've got our enzyme, we've got our substrate. Now, the enzyme can do two things. It can either break it down or it can build something. It can combine two things into one. Both of those are chemical reactions. And what an enzyme does is it makes those reactions go faster, okay? So if you think about it, if you were just to leave a banana just sitting out rotting on your couch or on your couch, why would you leave a banana on your couch? That doesn't make any sense at all. If you leave a banana out on your counter, over time it's going to get moldy and black and it's going to break down and decompose. What an enzyme does is it takes that reaction and makes it happen faster, okay? And how it does that is by taking the amount of energy that would normally require to start that reaction and lower it. Okay, so here we can see in this graph, the black line is the amount of energy that would normally be required. So in order to start the reaction of breaking down the banana, I would have to put in this amount of energy, okay? But what an enzyme does is it takes that amount of energy and makes it easier. It lowers it. So instead of having to put in this much, now I only have to put in this much in order to start that same reaction. And because I have to put in less energy, it means it can start sooner. And by starting sooner, it means it can happen faster. So enzymes lower the activation energy. Okay. Cool. Now, the reason that's important is because the odds of them giving you a enzyme graph or some sort of graph, like if they give you a graph, it's probably going to have to do with enzymes, okay? They're not asking you about evolution or ecology, so they can't ask you to like graph a population or graph a group of elk or something, I don't know. So that means if you're going to see a graph, it's most likely probably going to be an enzyme graph, okay? I don't know if you are, but it's probably going to be an enzyme graph. And the other reason I think that is because if they give you a specific graph and then ask you questions over that specific graph, that's not something you can Google. Okay, so they're not going to ask you like, how does an enzyme make a reaction go faster? Because that's a Googleable fact. You can just Google up lower the activation energy. But if they give you a specific graph that says this specific enzyme has this graph, instead of saying, how does an enzyme speed up a reaction? They might ask you, how much energy is the enzyme lowering the activation energy by? And you have to look and be like, oh, well, if this is like, let's say 100 and this is 50, well, it's lowering it by 50 units. Okay. So they might ask you to interpret the graph that has to do with enzymes, not necessarily ask you a specific fact. Okay. Now, because an enzyme is a protein, that means it's made of amino acids, and that means it's got all those hydrogen and covalent and ionic bonds that are connecting these things together. Okay. Now, if I wanted to break down those bonds, remember there's two ways that I can break down those bonds. The first way is with heat, because by adding heat to something, you dissolve bonds. Remember, those hydrogen bonds are really weak, and even covalent bonds are kind of weak when it comes to adding heat. I could also add in acid in the form of hydrogen ions. So I could take heat or I could take acid, and I could add that into this enzyme. And what it's going to do is it's going to take my enzyme that I've got here that's all folded, nicely folded and bonded together, and if it dissolves those bonds, it's going to go back to being my singular, oh, good Lord. It's going to go back to being my singular amino acid primary structure chain, which is cool. It's not fully broken down, but this strand doesn't do the enzyme job that I wanted it to do to begin with. So if it's in a primary structure, this can't break down that substrate, which means it's useless. It doesn't work anymore. Okay. And why that's bad is because when we remove the heat or when we remove the acid, the enzyme isn't just going to go back to being, oh, okay, well, let's just fold back the way we were. Oh, all right, cool, done. No, it's not necessarily going to fold the same way again. So after an enzyme gets denatured, some of them might fold the same way, but some of them will not, which is why you don't get exactly 100% function. Okay. So these are the two ways that we would denature an enzyme. Okay. You could also do it with any sort of with any ion that has a charge, okay, because those charges are going to disrupt our bonds. So anything that can disrupt a bond is going to denature an enzyme, okay? All right. So if I'm a betting man, which I'm not because I'm poor, if they're going to give you a graph, it's probably going to be enzyme related. So it could be something like this, right, where here's the optimal range for this enzyme, but now here's the new temperature, predict what the graph is going to look like. Here's the optimal range for this enzyme. Here's the optimal range for this enzyme. What pH do these enzymes work best at? Something like that. So this is something like what you could look like. Okay. That doesn't make any sense. This is something like what you could see on the test. There we go. And the last thing that we should talk about is inhibitors. Okay. Good old inhibitors. Now there's two kinds of inhibitors. Okay. So we have our enzyme, which I'm not going to draw with the big old muckled mess again. I'm just going to draw this. This is enzyme. What's a good name? I've used Ed before, or Eddie. Edward. Emmanuel. Emmanuel's too long. I can't write that in paint. Emmett. Emmett. Like Smith. There we go. Okay, so here is Emmett Smith, the enzyme. Now, normally we're going to have our substrate, which is going to go in there and fit into our enzyme, okay? But we have two types of inhibitors, okay? The first one is what we're going to call a competitive, because this inhibitor is basically going to be racing the substrate to the active site first, where if the inhibitor gets there first, this substrate can't bind, which means the enzyme can't break it down, which means the substrate is just out of luck, okay? Now, why would we want to inhibit an enzyme? Okay, well, think about it. If you eat food, you have enzymes that break down and build things. You have enzymes that build muscle. You have enzymes that break down muscle. You don't necessarily want all of those enzymes doing their job all the time. Okay, you don't want your stomach enzymes to always be breaking down carbs if you haven't eaten and there's no carbs for it to break down, because that's going to make you feel sick. So you want to be able to turn those enzymes on when you've eaten carbs and then turn them off once you've digested them all. Okay, so in order to turn them off, we have different inhibitors. Now this is a competitive inhibitor. Okay, where essentially it's racing the substrate to the active site. Okay, we've also got a non-competitive, where on our enzyme it's not trying to go to the active site. It's trying to go to another location. So maybe that location is here on the lower half. So our inhibitor is trying to bind to some other place on the enzyme, which causes the active site to change its shape. Okay, so instead of this being the shape, well, now this is the shape. So this substrate, when it tries to fit in here, it can't fit in because it doesn't fit in the active site. Okay, so the inhibitor is still inhibiting the enzyme, but it's just doing it in a different way. Okay. Both of these inhibit the enzymes. They just do them differently. This one binds, the competitive inhibitor binds to the active site. The non-competitor doesn't. Okay. We call this an allosteric enzyme. Okay. And we call it allosteric because when it binds, it changes the shape of the enzyme. So you can see here, when this activator binds, when the inhibitor binds, pardon me, it changes the shape of these enzymes to make the active sites no longer the same. Okay. All right, cool. That is a real quick snapshot of basically the entire first unit with everything that you fully need to know. Things that you have to know. Okay. So yeah. Be sure to like and subscribe.